Over the next few lessons, we're going to talk about concepts of the immune system, particularly concepts of innate and adaptive immunity, and we're also going to talk about pathogenicity, or ways that microorganisms cause disease. At the end of this particular lesson, you'll be able to describe the process of phagocytosis and how it contributes to immunity. You'll also be able to describe inflammation and how it contributes to the immune response. And then finally, you'll be able to discuss mechanisms used by pathogens to avoid the immune system. In particular, we'll talk about methodologies or mechanisms used by microorganisms to overcome phagocytosis and to overcome the inflammation that is part of the innate immune response as well. We're going to spend a few minutes with this slide here discussing inflammation, and in the couple of slides afterwards you'll see more of the specifics of what I'm talking about here. Though I'll include those slides, there won't be much additional talking points to them. So this is giving you a broad overview of the process of inflammation. The main role players in this process include resident macrophages, or macrophages that lie just under the skin in this example here, and then we've also got neutrophils, which will be recruited to the site of tissue damage, as will additional macrophages. These two cell types are what we classify as phagocytes, and we'll talk about the role of phagocytes in a few slides from now. So in particular, in a classical example of inflammation, we have some sort of tissue damage caused by a cut or an abrasion, which might lead to include bacteria or some sort of pathogen on it, and that bacteria then gains access to the tissue below the epidermis. What we find is that the bacteria that gain contact here then are going to interact in some way with the macrophages. The macrophages will phagocytize them and then begin to secrete what we call cytokines. And the cytokines have numerous different roles. Some of the roles of cytokines are to activate other immune cells like neutrophils and additional macrophages that are within this site of where the infection is taking place. Some of those cytokines, as shown here, might diffuse within the bloodstream, in which they begin to recruit neutrophils and additional macrophages to the site of infection. In addition, we find that the cytokines can contribute to changes in the architecture of the epithelium that line the blood capillaries. In this example here, what you're observing is that these cytokines will actually bind to the surface of these epithelium, and in doing so will cause these epithelium to loosen their tight junctions that hold them together, allowing for not only eventually white blood cells to make it from the capillaries into the infected tissue, but it also allows for the movement of fluid, plasma in particular, from the capillary into the tissue as well. This movement of fluid is known as edema, and the dilation of the epithelium that occurs throughout this process, again driven by those cytokines, is known as vasodilation. This not only will increase blood flow, but also leads to some of those hallmarks that we just described, such as that loosening of the tight junctions between those capillaries, or between those epithelial cells within the capillaries. In the third step of inflammation, what we find is that neutrophils, as they become contacted, or as they come into contact with cytokines, they begin to actually undergo a change. Before coming into contact with cytokines, neutrophils kind of just roll their way through the blood vessels. But once they come into contact with a cytokine, it causes a change in the cell, and where it begins to actually crawl along the capillaries towards higher concentrations of cytokines. This movement towards a higher concentration of a chemical is a term that we've seen before. This is the process of chemotaxis. And the ability of these neutrophils, or when these neutrophils begin to start crawling along the surface, as is shown with this one right here, this is a process known as margination. Finally, we will find those neutrophils squeezing in between the epithelium as the plasma was able to move through the epithelium. And in this process, this is known as diapedesis. Diapedesis. And this allows for the neutrophils to continue moving towards higher concentrations of the cytokines. And those cytokines, again, the point source of them is this macrophage that has gobbled up some of the pathogen here. And this then allows for the recruitment of neutrophils to that site where infection has occurred. Now, neutrophils are the first responder. They will respond within minutes to some sort of tissue damage. Macrophages will be recruited as well, though their recruitment takes a little bit longer, more on the order of about an hour. And the last step of this process then, what we end up getting is destruction of the pathogen, but also we can find the formation of a blood clot and also possibly an abscess if the infection isn't totally cured or totally healed within the first few hours. Here we're going to find some bacterial cells, macrophages, neutrophils, dead cells that have died from damage from the infection that may have occurred. But over time we find that these abscesses will resolve themselves and that cut new skin will be regenerated at that cut site, allowing for healing. In the next couple of slides, you will see just a recap of what I've discussed. I'm not going to include any additional comments to them, but this will provide more of a step-by-step process of what's occurring in the inflammation process. Now, when we talk about inflammation, we often associate it with a number of signs and symptoms, including redness, swelling, warmth, pain, and itching. And though some of these are caused by the vasodilation and edema that occur during the inflammation process, histamine can contribute to some of the pain and itching. And histamine can be secreted or released from eosinophils, which are another type of white blood cell, and might be also secreted by neutrophils during the course of inflammation. In broad speaking terms here, all of these different signs can be attributed to cytokines, which are secreted during the inflammation process. We mentioned in the past couple of slides the hallmarks of inflammation, and one of the things that we mentioned is that during inflammation, neutrophils and macrophages will phagocytize pathogens. In the next few slides, we're going to talk about this process of phagocytosis and not only how this leads to the destruction of microorganisms, but then how this also actually enhances the immune response. We will also talk about in a later lecture that B cells can also perform phagocytosis. And what I'm providing you here is an image of a macrophage in this orange color, and in the golden color are bacteria that are being actually attached to the macrophage. And you can see in this particular example, this macrophage, or this specialized macrophage known as a dendritic cell, have these really long extensions or arms, which allow this, and the amount of surface area here allow for contact with the bacteria. These arms would then retract, and eventually what we will find is that the macrophage will eat or phagocytize these bacterial cells. There are 5 major steps in phagocytosis. Attachment of the pathogen to the macrophage, the formation of a phagocytic vesicle as that pathogen is brought into the cell, maturation of the phagosome, in which we find possibly a decrease in pH, Also, we will find at this point then the fusion of a phagosome with a lysosome to form what we call the phagolysosome. And during this fusion, not only do we find an increase in the drop of pH, but we also begin to find the production of nitric oxide and other reactive oxygen species. And you may recall from some of our past conversations that the production of reactive oxygen species can be quite damaging to bacterial cells, particularly those that don't have catalase. Well, this oxidative burst, or this phagocytic burst, which is in part driven by this enzyme phagocyte oxidase, is critically important to the destruction of microbes within phagocytic vesicles. What we find is that these reactive oxygen species and nitric oxide will end up essentially damaging the cell and leading to the cell's lysis. We'll talk in a later lecture about what happens after this process to the remains of that bacterial cell or to that pathogen in general. But in the next couple of slides, what we're going to cover are a couple of very specific points. One, how does this attachment occur? And secondly, how does the cell respond about this event to other cells after phagocytosis has occurred? You may recall from our conversation on viruses that for a virus to attach to a host cell, the virus has to attach to some sort of receptor on the host cell's surface. So here is a receptor, here would be like a spike on a virus, and that allows for the attachment. Well, when it comes to phagocytes, particularly macrophages and neutrophils, we also have to have some sort of attachment between the host cell, the macrophage, and the pathogen. And the attachment here can be driven by a number of different proteins, which we call pattern recognition receptors, or PRRs. PRRs are a very broad class of proteins that recognize hallmark characteristics of pathogens. For example, you might recall that all gram-negative cells have lipopolysaccharide. That lipopolysaccharide is a generalized pattern that is recognized by PRRs. Lipopolysaccharide, peptidoglycan, and teichoic acid on gram-positive bacteria all represent what we call pathogen-associated molecular patterns. And these generalized patterns are what get recognized by those PRRs. A specific type of pattern recognition receptor is known as the toll-like receptor. And these toll-like receptors, I'll give you a couple of examples of what these recognize in the next slide, but these were the first of the pattern recognition receptors to ever be identified and associated with immune response. Now what we find is that the binding between these pathogen-associated molecular patterns and the PRRs will not only allow for phagocytosis, but also trigger a number of changes within those cells in which we observe this binding, and those changes can lead to the secretion of cytokines, or chemoattractants known as chemokines, which are a subset of cytokines. And the secretion of those molecules can ultimately lead to the inflammation response. Here are just a few specific examples of pathogen-associated molecular patterns that we might find in viruses, bacteria, pathogenic fungi, or pathogenic protists as well. And what you see is mostly abbreviations here, LPS being the lipopolysaccharide on gram-negatives, LTA being lipoteichoic acid on gram-positive bacteria, and then beneath these you can see as well the particular toll-like receptor that recognizes each of these various pathogen-associated molecular patterns. You'll also see that DNA and RNA are actually associated patterns, molecular patterns as well. And we're not going to go into the reasoning of why they particularly are considered PAMPs, but if you are interested, please feel free to ask. So in the past few slides, we've talked about the importance of phagocytosis in destroying microorganisms, and that the attachment of a pathogen to a phagocyte is an important part in that process. Finally, I want to point out that these phagocytes, like macrophages and neutrophils, actually signal or tell other cells that this event has occurred. When macrophages or neutrophils ingest a pathogen, they become activated, and in part the activation leads to the secretion of various cytokines. This image here is just showing you an example of some of those cytokines, and I am not right now asking you to know specifically what these cytokines do, but I do want to point out a couple of things. One, some of them are chemokines. They're recruiters that attract specific types of cells to the site of infection. Some activate inflammation. Some will lead to vasodilation and the edema that we observe. And then finally, not shown in this picture, but there are some, such as IL-10, which eventually signal to turn the immune response off, or to turn off inflammation. And if we do have time, down the road we'll talk about a viral mimic of IL-10, which actually inhibits the immune response, therefore benefiting the viral life cycle.